Hello, my name is James Schnabel, and today I'm going to be talking about how to use GEvo for people who don't have previous experience with it. GEvo is part of the Koji toolkit, and I'm actually making this video because of a new resource we've just made available showing classical maize genes and equivalent model in the reference sequence of the maize genome that was released by maizesequence.org. So as you can see, here's the spreadsheet, which you can also download as a CSV file if you want to use it on your own computer, with the gene symbol and then the maize sequence that it maps to. And the other thing I want to draw your attention to is over here we also have tried to identify orthologous genes in various other sequenced grasses, as well as two regions in maize. You'll remember that maize has gone through a whole genome duplication sometime in the last 5 to 12 million years, so there are now two regions of the maize genome that correspond to every one region in sorghum, rice, or brachypodium, the other sequenced grasses. So if you're interested in this data, let's say for a gene like C1 or colored alurone, you can scroll over here and you can see that colored alurone is, let me make sure, okay, it's the first one in this case, so it's in maize 1, and here is gene model that was assigned to that gene by MaizeGDB, and here you can see this is what we think the orthologous gene in sorghum is, and here we've got an orthologous gene in rice. However, in brachy, this is not an actual gene model. What this is the region that we predict to be orthologous to where we find these other genes, even though we weren't able to find a gene that corresponded to C1 at that location. So to show you what that is, I have to take you to the GEvo panel of these regions, which you can get from clicking on this tiny URL link. It's listed as the syntenic ortholog panel in the spreadsheet. So this can be a little overwhelming if this is your first time ever working with GEvo, so let's walk through it one step at a time. What's on the screen now are 5 different panels. Starting at the top, this is the sorghum panel, the maize 1 region, which you'll remember contains C1, the maize 2 region, which in this case contains a homologous gene, which in the case of C1 I believe is actually the PL1 gene, which is also a known, well-studied maize mutant. Then we have rice, then we have brachypodium. Within each panel, you can see these little green and gray arrows represent known gene models. So genes facing on the top are genes that face forward on the strand, and genes on the bottom are simply in the opposite orientation. And the other thing your attention will be drawn to is these colored boxes, which represent blast hits to one of the other regions in the panel that we've looked at. So if I click on one of these, for example this, you can see we get a pop-up box with a great deal of information about the blast hit. And if we want even more information, we can click on full annotation, which brings up everything about the sequence, and you can even see the alignment here if you are so interested. But let's disregard that for now. Get back to the actual panel. You can also see that you can click on this and see the where each hit maps, which is useful. You can do a shift click and you can see all the matches between any two sequences. Let's clear those. If we're interested in seeing where sorghum matches to the other region of maize, we just click on the second one, like so, and you can see now it's showing us which hits map up between this region in maize and this region in sorghum. So the other thing to note here is that you can see these also have an orientation. If the two blast hits are in the same orientation, they're printed on the top. If the two blast hits are instead in the reverse orientation, so forward to reverse strand, they show up on the bottom, like here. So you can see this one particular gene appears to be inverted in brachypodium. So here is the ortholog, syntenic ortholog, as far as we can tell, of C1 within rice, and it's also the syntenic ortholog of PL1, remember, because they duplicated in maize but not in rice. And you can see that this does not have a hit to brachypodium. Brachypodium, as the bottom panel, would be the bottom layer of colored boxes here. So if we click, do a shift click to see all of the hits, you can see that all of these genes line up very nicely, and all of the genes over here line up very nicely. But this gene appears to be completely missing, although there is this chunk of purple here. And you notice that a lot of these panels do have a great deal of purple in them, and the reason for that, by default, we mask all of the non-coding sequence if we find the gene. If we don't find the gene, we don't mask all non-coding sequences, but we do mask repetitive sequences. So in this case, it's possible the gene is hiding under the masking. So what we'll do is we'll go down to our controls here, and we can change from the masked version of brachypodium to the unmasked version. So then in rice, what I will do is no masking on the CDSs. So you can see we have several options. We can mask everything that isn't in a gene everything that isn't a coding sequence, or we can mask everything that is a gene, which is useful for other purposes. But in this case, I'm going to ask for no masking in rice, although we'll leave the 50x repeat filtered version of rice in there. So I run the GEvo analysis now. So now you can see that these genes are still being mostly filtered out, which to me indicates that they're probably transposon-like sequences. And when we click on this, we can see that Tiger has already annotated this as a putative cacta transposon. But still, the main thing to note is that we still have these two hits in brachypodium, and these are the sort of hits we use to predict where a syntenic region is, but the C1 gene appears to be completely missing from the sequence, which is something that probably bears further investigation, but not in this video. Anyway, I hope that this has been an interesting introduction to how you can learn more about your favorite gene using syntenic panels of it made available in the classical Maize gene spreadsheet, and using our tool GEvo.